This is TWIS, This Week in Science, episode number 1017, recorded on Wednesday, June 4th, 2025. Just banging rocks on trees. Hey everyone, I'm Dr. Kiki, and tonight on the show, we will fill your head with water tanks, tiny violins, and garbage gulls. But first, thanks to our amazing Patreon sponsors for their generous support of TWIS. You can become a part of the Patreon community at patreon.com/thisweekinscience. Disclaimer, disclaimer, disclaimer. Don't let your confidence waver. Although this bump in the road looks like a monstrous cane toad, this moment can be ours should we meet it. And if you despair, remember we're always here at This Week in Science. Coming up next.
I've got the kind of mind that can't get enough. I want to learn everything. I want to fill it all up with new discoveries that happen every day of the week. There's only one place to go to find the knowledge I seek. I want to know what's happening, what's happening, what's happening this week in science. What's happening, what's happening, what's happening this week in science.
Good science to you, Kiki.
And a good science to you too, Blair, and everyone out there. Welcome to another episode of This Week in Science. We're so glad that we could be here to talk about science with you. It's one of our favorite things to do. I mean, I don't know. I'm talking for myself. Blair, do you like it?
I mean, it's
Yeah,
up there
okay.
for sure.
It's up there.
I mean, I choose to keep coming back for the last 13 years, so
Just a bit.
I must enjoy it.
Yeah. Yeah. We do enjoy it, and we're so glad that any of you who have been coming back over and over again still enjoy the show. And that I do hope that new people come to the show and decide they want to come back and back and back and back, just because it's a really good time. On tonight's show, I have stories about a light limit, a tiny violin, a garbage gull, making myelin, and something about mouth tape. You should say no to it.
That sounds correct. Is this me playing the smallest violin, Kiki?
It could be. I mean, it's
Okay.
a very, very tiny violin.
Okay. I'm excited to hear about that.
It's very exciting. Blair, what is in the animal corner?
Oh my goodness. I have some amphibians, wet as they usually should be. Yes, I have late breaking trash parrot news. That's what I was looking at a moment ago. I have orderly baboons and flighty climate change. Oh, and also Kiki and I are going to talk about chimps a little bit.
I like chimps.
Yeah.
Blair likes chimps.
Yes.
Yeah, so we're going to talk about it. Yes. I love
Lots and lots of stuff.
it. Yes. I'm so excited. I hope everyone here is excited for all these stories and more, and that you are subscribed to the TWIS podcast everywhere that you can find us. We are pretty much on most all of the podcast directories that are out there, and we are on Facebook, YouTube, and Twitch, live streaming Wednesdays at 8 p.m.-ish Pacific time every Wednesday evening, and other times when we kind of feel like it. And if you subscribe to those video platforms and get the notifications, you'll find out when that is. But, the podcasting, that's great. If you want to find out more, like show notes and other stuff, you could go to our website, twis.org, and find out the things that are there. Follow links to subscribe to the show. It's all sorts of fun stuff. That's where you get the Patreon community, our Zazzle store, all that kind of stuff. It's really great. Are you ready for the science, Blair?
I'm ready. Bring it to me.
I will. I will serve it up on a platter of disenchantment. Okay. There's been a lot of news this last week coming out about what really is going to happen to the future of American science. We have the big, beautiful bill that passed the House and is sitting in the Senate right now and being debated. And it currently is set to massively reduce science funding across
Mhm.
the institutions within our government. So National Institute of Health, National Science Foundation, NASA, NOAA, CDC, you name it. These organizations are being gutted. And for like a general sense, it has been calculated by people, and this is a post on Blue Sky, but that the numbers would take us backwards 25 years
That
in budget.
seems low, honestly. I think that, I mean, so yes, there's the initial take back, but then I feel like there's the lag in progression that means we're going to go back 25 years. So we're going to be in the year 2000, and then we won't progress for the next 4 years. So we will stay 25. So then we'll be 30 years behind. Like, I think that's the two pieces, right? There's like the rollback, and then there's the lag of return.
And I think it's not, so this 25-year rollback of R&D is not the same as the educational pipeline, the talent pipeline, the funding that is being cut to grant programs, training programs, efforts to bring more people into the sciences to actually benefit American scientific research and development. So, yeah, you're right. The timeline, although these cuts will pretty much take us back 25 years, we're also going to have the lag because of the talent pool and bringing development back up, manufacturing back up, our ability to actually compete and to get collaboration with international scientists, to collaborate on national projects like the International Space Station, which this week it was reported that NASA, because of budget cuts, has pulled out of the annual International Space Station Research and Development Conference, and additionally, another conference that's an international conference for astronomy and space science that they fund. And so these two conferences that are international conferences where NASA, it's an opportunity for NASA to go, look, world, look what we're doing. Maybe you should launch stuff with us. Have contracts with us to do stuff. They're not doing that. And so where's it going to go? Who is going to pick up the slack? How is this? Yeah.
I thought space was one of the things that like this administration was all into because of Elon, but
yeah. And so this is very interesting. Another bit of NASA information. One of Elon's cronies, Isaacman, the businessman who funded one of the recent SpaceX launch that did the high altitude evac maneuver to test new astronaut suits. He was in line to be the head of NASA, but Trump and Elon got in a fight, and so that didn't happen. And everybody was kind of okay with Isaacman because he's really pro-space and everything. I don't know. No, I don't know exactly what I said that, but I don't know exactly what went into that decision. So there are going to be other people being considered for heading up NASA. We've got a lot of issues with NSF. NSF, its budget is going to be potentially reduced by 57%. So from a little over 9 billion to 3.9 billion. And all of it is pretty much from getting rid of STEM training opportunities. So scholarships are reduced, a 65% cut, postdoctoral fellowships, a 91.4% cut, NSF research traineeship program completely eliminated, broadening participation initiatives, cut 1.6 billion to 171 million, and other programs, zero being cut, are advancing informal STEM learning, CISE education and workforce, research experiences for undergraduates. And we're going to see, though, a program remain that's the Tribal Colleges and Universities program, HBCU Excellence in Research, some other programs, but massive budget cuts. So the TCUP, which aims to increase the number of Native Americans in STEM careers, will receive 7.1 million compared to 16.5 million. Additionally, NIH is going to be severely cut. Indirect costs at universities capped at 15%, which is going to limit the ability of universities to support research initiatives and labs with equipment that could support collaborative opportunities. So there's a lot going on here that is going to significantly and severely reduce American potential for our future, for our scientific advancement. And yesterday, Marcia McNutt, the president of the National Academy of Sciences, made her second annual State of the Science presentation. She said, "Oh, look, haha, I didn't think I'd have to do another one of these, but I guess I do." She basically said, "Hey, everybody, there's a lot going on, but what we need to do is not erode America's trust in science anymore. We need to work on building trust, building communication, and push our ability to remain a world leader in scientific innovation." She says that we need to build a culture of innovation, so budget cuts are not going to help that. We need a national research strategy involving basic and applied research so that there's data-informed analysis that leads research efforts to maximize impact. So strategically planned impact, saying that countries like China, Germany, Japan, and Norway do this kind of strategic planning with apolitical non-governmental organizations with, consistent funding as opposed to funding that is from year to year and congressionally allowed.
Yeah, apolitical government organizations, what are those? We don't have those anymore.
Well, supposedly the National Academy of Sciences is supposed to be one of those, where the National Academy of Sciences is external to the government. They do get governmental funding for their research and reports that they do, but they have to get external funding to maintain
right.
but they are a congressionally mandated organization of scientific leaders, of academics that are supposed to advise Congress, that are supposed to advise the government. That's the National Academies, everyone. That's what it's supposed to be doing. She said we need more international collaboration. So, hey, don't cut those indirect costs. We also need, she said this, that red tape regulations need to be reduced. So there's a lot of reporting, there's regulations involved in NIH, NSF grants, and a lot of these governmentally funded research programs that impact the ability of researchers to actually do research. So that researchers are spending a lot more time doing paperwork and making sure that formatting is correct as opposed to actually doing work that would be valuable.
Yes.
And it's true. But I think along with this, as a science communicator, I would say that the government should be prioritizing science communication funding so that there is NSF, NIH funding for budgets, for universities, for researchers to hire professional science communicators to do communication work for them when needed, so that that's taken care of. And it's another bit of stuff that is off the researcher's plate. That's what I think.
I think it's so strange. Obviously it's being heavily normalized, all these strange things that are happening all around us. But I just think it's so strange that funding is being severely cut to things that no one in the history of American politics would ever consider cutting funding to, because you're physically doing harm. You shouldn't physically be doing what feels like intentional harm to other humans via legislation, and that shouldn't be allowed to happen. And the idea that you're like, no more cancer research, it's fine. Like, that's a crazy thing to say. We're spending too much money on cancer research.
Cancer research gets to stay. I think that's one of the ones that gets to keep its funding. But there are like three main areas that get to keep their funding. It's like cancer research, nanotechnology. I don't know. It's interesting.
Okay, well, anyway, but yes, disease in general, right? Like, even if you want to take all the other science out the window, but like disease-based research, things that will directly save lives, cutting funding to those is wild.
But, we're also cutting funding that involves, invasive species, invertebrates.
Oh, of course. How about climate change? All of the climate change research. Yeah. All of the climate change communications funding. Noki lost a huge NSF grant. Like, it just,
It's a big sea change, and I think it's very interesting. And I will caveat the National Academy of Sciences report or presentation with the information that the National Academy of Sciences has been in the red for the last 2 years by significant millions of dollars, and is probably going to have to cut some 250 people very soon, and cut programs of their own because of the changes in funding and, congressional or administrative wants. Priorities. So it's an interesting time to be doing science and be a science communicator and wondering where this all is going. And on that note, what I will say is that this is in everyone's power to have a voice on. The big, beautiful bill is not yet passed, and you can call or contact your representatives, your senators. 5calls.org is one great place to go. There are places you can go. You can make your voice heard and have a voice. It may influence. And very often the senators, they're just waiting to hear from the people that elected them. And if there's a big enough clamor, it might just change things, because I think there's enough happening right now that if people spoke, if people said things, that
it needs to be more though than it's ever been before, because for some reason
every day, 3 times
the politicians are not listening in the same way that they have historically. There's different pressures, there's different things happening that are making Congress make weird decisions, and so there is an opportunity to sway them, but you have to try harder, harder. and there are scripts, and like you said, you can go to 5 calls, they'll give you a script. You can go to other places where they'll give you a script. You don't even have to come up with what you're going to say. You can
write a letter to the editor in your letter to the editor in your local paper. Try and talk to people. This is not about progressive lefty politics, even though we're talking about politics. What it's about is maintaining the research, development, and economic future of this country. If you destroy the knowledge creation engines, universities, labs, if you destroy those, your ability to compete, to create, and to advance goes away. So,
I think a lot of these initiatives too that are being that are on the chopping block were started by Republican administrations, right? So that's the other thing is a lot of things are being thrown. It's not a right-left situation. It's like a sense and decency and no sense and no decency. It's insane that Reagan himself would not have made these cuts. It's wild.
No, and so, this is what's fascinating. This is also the 75th anniversary of Vannevar Bush's creation of the NIH, NIS, NSF, and his endless frontiers philosophy. So Vannevar Bush, the first Bush, he came out with this big thing, the endless frontiers, and we're going to be a scientific powerhouse. And he created, with a lot of other people, created the beginnings of what has gotten us to where we are now. And it's fascinating to see that being erased in a moment. Yes, changes need to be made. Yes, there's a lot of institutional issues, But what's happening right now? What? It's gonna take us so long to come back from this, you guys. Come on. Call people. Write letters. Let's do it. Not just Blue Sky. Blue Sky is now where you want to be. Facebook. That's now where you need to write letters. Talk to people. Call people. Yeah, Nixon gave us the EPA. Thank you, Joint Specialist. Yes, exactly. We got to do it. I mean, that is the thing that I think the reduction in civics education in our schools has left us with is a disenfranchised public. People do not think they have a voice anymore. They don't understand how they can impact things. So civics, you're a part of this. You are part of our body politic, right? We're part of it. So let's do something. Okay. Okay. Now that was it. That's all I want to say for that right now, because we can do this and we don't need to get all upset about it. I mean, or if you need to get upset to do something, then get upset. But like, seriously, come on, buddy. Write your senators. Come on. Call your senators. Blair, let's talk about like actual science. We're going to move on now, everyone. What do you want to talk about?
That was actual science. All right. Anyway, let's talk about some newts and frogs and toads and salamanders.
Okay. What you want?
So those are amphibians. Amphibian means double life or something like that. I don't know. I haven't worked at a zoo in a while. But ultimately, no, their name has to do with the fact that they live on land and in water. They need to stay wet. They do not have watertight skin. A snake or a lizard, reptiles have watertight skin, so they can live in the desert. A newt cannot live in the desert. So all that to say, they need to stay wet. And so especially in climate change, in periods of drought, amphibians are impacted heavily. Also because they breathe through their skin, they're considered indicator species for pollution because they are impacted very heavily by any contaminants in the water. All that to say, amphibians are something to look at when you are studying how an environment is doing, how an ecosystem is, how healthy it is, right? And so in Portugal, they found that amphibians were spending a lot of time in historical irrigation channels.
Ooh, I would too. That sounds
Yes.
great.
So this is hundreds of years old stone water tanks and irrigation pathways that were used by traditional villages, and now they are critical breeding and refuge sites for amphibians because the natural wetlands are under a lot of pressure from climate change, from use, and from contamination. And so, yes, so they were doing kind of a general amphibian population survey, and they were able to look across 162 bodies of water, which included natural sites like ponds and streams, but also included artificial structures like tanks, fountains, and irrigation channels, all made of stone. They looked at species richness, abundance, presence, and breeding activity. And what they found was that while natural streams and ponds supported a greater number of species, the artificial water bodies had an unexpectedly vital and complementary role to the natural bodies of water. So specifically the stone tanks had extremely important breeding potential for amphibians. A lot of them were breeding in these tanks. And they even supported a higher diversity of species compared to natural habitat types in the tanks. Specifically, the marbled newt and the Bosca's newt were breeding in these historical structures more frequently than in nearby ponds and streams. That being said, they didn't replace the ponds and streams. There were some animals like the Iberian frog and the fire salamander, which only were found in natural water bodies. They weren't scooching over to the historical sites. But this is a case where the historical site has to be included in the assessment of the space and is important ecological territory. And so what this reminded me of actually is when a shipwreck becomes a coral reef.
Yes. Okay. Yeah.
So
When
that
does
is a
the
man-made
artificial
structure.
become-
Yes. Yeah.
Yeah.
It's an artificial structure that feels like it shouldn't be there. It has disrupted the natural space, but over decades or centuries has become a habitat space. And so, however the historical communities diverted water into these areas, that has maintained, and they have been able to use this as fresh water for actual amphibian habitat. And so it's important to know this when you're doing ecological studies in general, studying species health, but it also is important to know that you have to protect those man-made structures alongside natural habitats, because the natural structures, not only are the man-made habitats now in their own way natural structures, but the habitat itself, the ecosystem, is different than it looked hundreds of years ago. So due to human impacts on the space and climate change and drought and pollution and all these other things, some of these species might have disappeared or moved, but they did not, because these man-made structures exist. And so that is essential to the assessment of the space and the species.
Now I want to know why. Like, why do some of these man-made spaces become protective for some species? Why are they great? And then not for others, like the salamander, and they're like, nope, I'm going to be in the natural habitat. That's it. That's the only place I can be. Yet others are like, yeah, whatever, it's good.
So I could wager some guesses, right? Just based on what I know about amphibians. For example, some of them might require more depth in their water to breed. And the natural habitat might not be able to provide that depth because of erosion or drought, right? So then that's why they might prefer the historical man-made structure. The animals that are not venturing towards that might not be able to go as far. And so they might not, over however many generations, have been able to make the adjustment, because also a lot of animals aren't staying in those spaces. They're laying their eggs and leaving.
Yeah. What is it? Is it the tiger salamander? It's one of them where the salamanders, they're born in one little vernal pool, and then they crawl through the undergrowth of the forest. Basically, they search for another vernal pool, and so they're on a journey. Right?
Yeah. And so some of that could just have to do with the radius. How far away are these historical structures from the pools that they're born in?
How many streets do they have to cross? Yeah. This is like not Frogger, salamander. I don't know.
Right. Is it a newt that really needs to stay pretty wet? Or is it a salamander that can walk for a couple days in moist soil and they're wet enough? It all depends on that kind of thing too, for sure. Also, what do they eat?
Yeah.
Is there food over there in the historical spaces, or is it not?
Okay, this is something I was thinking of. So these historical spaces, though, here in the United States, I'm looking at some of these little pools of water. They look like they would grow mosquitoes really well. And like in a lot of communities here in the US, it would be like, get rid of it. We don't want the mosquitoes, right? Or
if there's enough frogs and salamanders,
right?
Yeah, absolutely. And if there's enough amphibians, maybe it's not that bad, the mosquito problem. Maybe laying eggs in a pool that's going to have mosquitoes in it means your babies are going to have plenty to eat the second they hatch.
Okay, so now what I want to do is take a whole bunch of amphibians to Canada.
Yes, absolutely. Take amphibians everywhere, except for cane toads. Don't take cane toads.
No, and I'm not going to. No, we're not going to do any kind of movement of biological species.
No, just breed the natives and release a lot of extra native frogs. That's not going to hurt anybody.
But it's an interesting balance, right? It's like we don't know, and especially in all of these spaces where this study is learning about this thing and is saying we have traditionally ignored these spaces as habitat because it's urban.
Right.
And so we need to learn more about this, but this is also at the time where climate change is occurring. A lot of these spaces are getting hotter more of the year. There's also, so across Europe, you have changes to these areas where the summers are longer and hotter. They also have changes in mosquito movement. But how is everything moving? Who's keeping up with whom? And are they? I don't know. I think this is a really interesting beginning. I don't know. it's fun to come in and get a snapshot every once in a while.
Yeah. And I mean, once again, talking to the people who live in the space where you're studying native species is really important, because I don't know this for a fact, but I would bet that people who live in this area in Portugal know that these historic pools are full of newts, right?
Sure. Or at least all the kids do.
Yeah, oh, absolutely. And so you would expect that they would be aware of that. I don't know if researchers would know to check there, right? And so I think that's the other piece of this, is making sure that you're speaking with native populations who know the history and also what's the bigger picture of what's going on. They're not going to show up with their PVC squares and just kind of do their survey and then go back home. They're living in that space, and so they have a more intimate knowledge of what's going on.
Yeah, I mean, I hope that all research is incorporating communities these days. People-powered science. Come on, everyone. Speaking of powering science, artificial intelligence is doing so much these days. And this is actually a cool story. This isn't the chatbot kind of stuff. This is algorithms, models based on neural networks. Researchers at TUVN and the University of Glasgow and the University of Grenoble, yeah, American accent. They have been trying to figure out how can they make the best microscopes. How can you make a microscope as accurate as possible? And similar to how telescopes have to deal with diffraction and issues with the light that's arriving at the detector in that telescope, microscopes are the same way, but instead of, it's enhancing, right? You're measuring something that is very, very small with microscopes, but often you're looking at cells in a tissue. That tissue is maybe opaque. Maybe you need to differentiate different organelles from background stuff. And you're going to be dealing with the limit of optical measurement technology, the limits of optical systems to be able to actually detect light. And so these researchers publishing in Nature Photonics this week have published their work where they basically came up with a way for people who have these microscopes, which now they call deep learning microscopes. Deep learning microscopes are algorithmically based, and you can teach them things. So you give them a lot of information. Look at this picture, this picture, this picture, this picture. And because of the algorithm involved, it's able to, and in the case of neural networks, it's able to make correlations and connections that are not just straightforward. So the researchers here, what they've done is they took a microscope and they took a laser beam and they shot it through a liquid to hit a mirror so that that light then would bounce back, go through the liquid, which is where the microscope was like looking. And then the light from that liquid would then emerge, be refracted, and would get to the microscope. Now, the microscope is just getting images. The algorithm, the neural network, is what is being trained image by image by image. And so they slowly increased what's called the turbidity, which is the opacity, the random chaotic mix of stuff in the liquid, so that as that laser reflection bounced back through the liquid, it would be refracted in different ways. And it's a chaotic progression. And so this algorithm is learning how it sees things at a certain distance through a certain material. And it was looking to see, basically the researchers were like, we're going to tell the microscope to try and figure out where that reflected light is coming from on the mirror behind that liquid. But because that turgid fluid became thicker and thicker and more chaotic, it got harder and harder and harder because it's like having an opaque filter in front of a light and that light gets dispersed. When we look at it, it's like, oh, it's a fuzzy light. We can't actually say there's a laser beam being reflected from back there. It's a pinpoint of light, a point source. We can't say that. But a microscope with an AI algorithm, I'm sorry, I keep going to whatever the modern media is doing because I read it all the time. This neural network algorithm trained over and over and over to learn how it sees and to understand. I mean, I'm using anthropomorphic terms, but to understand the limits of its detection of light. Now they can take this microscope with this algorithm and go, okay, we're going to put it in front. This one microscope that we taught to see through stuff this way, we're going to have it look at a slide that's got, it's this thick. It has this tissue in it. We're going to tell it to look at this distance at this thing. And we're going to train it to do that. But now this algorithm knows its optical limitations. And so in this study, what they really showed is that they were able to almost reach what is considered the theoretical limit of optical detection and measurement. And so this, because it's almost the best that any because of the algorithm, because of the learning, the detection can potentially make other microscopic systems all over the place way more accurate. So it's kind of an interesting thing. So we might get better optical detection, which could help us see cells better, help researchers understand how things move in fluids or in tissues, be able to do all sorts of measurement and detection. Yeah, it's kind of fun.
Interesting.
Do you really think so?
I'm having trouble understanding why the fluid. I just still don't understand that. Why the fluid?
Oh, why did they have to?
Is that because that's how an eye works, or?
So if you imagine imagine that you're looking at an aquarium, right? Mm-hmm. And there's maybe a fish in that aquarium that you want to see, but the aquarium is dirty. Mm-hmm. And there's a bunch of particulate matter in the water. And we cannot see that fish necessarily or detect the colors that may be imaged, but the light bouncing off of that fish and making its way out through that turbid water, because of the way that light bounces and refracts off of different different material or different elements in solution, different molecules in solution. The diffraction gradient, the refractive index of the fluid that the detector, the microscope, the neural network that's trained could know how light moves through that fluid and actually kind of control for all that noise.
So they'd be able to see the fish.
It would be able to potentially see the fish. Yeah. I mean, I'm making this kind of a more macro thing. And
no, I appreciate that. Yes, I understand. It's like, it's teeny tiny. It's not fish sized. I understand.
But it could be all sorts of things. So it's using, the idea is that by having, like if you watch an aquarium get dirtier and dirtier and dirtier, if you were to have a neural network observe that aquarium get dirtier and dirtier and dirtier, but have had a control, and then also all the samples of looking at light bouncing through that fluid as it got dirtier and dirtier and dirtier, that neural network could then model how light moves in that fluid and refracts within it, and how and understand what it sees better than our brain.
Okay, and so give me one more. Why? Why do we need this? What is this for? Is this just to test the limits of sight?
No. So this actually will help us do all sorts of tissue sampling better and faster.
That's what you were saying. Okay, I get it now.
So instead of having, yeah, I mean, we'll have to have people do control observations and measurements for a very long time. But right now, it's like you can have an algorithmic microscope basically say, I see this mitochondria in this tissue over here, and we're going to do it. And if you have a tissue that you sliced a whole bunch of times or whatever, that algorithm could then actually give you information that is specific to that one particular mitochondria. And it doesn't have to be a graduate student who followed that mitochondria through that particular sample. Yeah. Oh my gosh.
Okay, so it is interesting. I'm just like, it was very theoretical still to me. So I get it now.
And I mean, thanks for asking the question because, you know, in my head, I was like, I think I understand it, you know, and then, you know, but you got,
it's for tricorders. Absolutely.
Yeah. Misha Baroque is saying, I wonder if they could use that yellow dye that they make Cheetos from that makes you see through your skin. I actually asked my husband this exact question earlier. I was like, why do we need an algorithm when we have yellow dye number 5? Come on. Waka, waka,
That's
waka.
right.
Yeah. Oh my gosh. But anyway, yeah. And this is not one of those, I think, scary things. I think this is actually improving our ability to detect and measure. And so by making things more accurate and I think it's really fun and could have the potential to give us lots of information and save lives.
Less AI for ads and scams, more AI for healthcare, please.
Yes. AI to help science do science. Do the science. That's what I want. I want algorithms to help do the work.
I want algorithms to find cancer a year before we would. That's what I want.
Yeah. There was some news out this last week, I think, or recently that detection algorithms where doctors or people would go, oh, there's cancer in that, or, you know, trying to detect, like, people are still slightly better than the algorithms more of the time. But algorithms are also starting to become very, very good. But I don't know, who else is becoming good with lots of practice, Blair?
Oh, parrots? Is that what we're talking about?
Maybe.
So some of you keen-eared listeners will remember the trash parrots. This is in Western Sydney in Australia. There are cockatoos who have figured out how to open trash bins to scavenge. They're sulfur-crusted cockatoos. And they're so good at it that they keep outwitting the humans who are trying to lock up the cans, creating what scientists describe as an innovation arms race. And that's what I reported on, I don't know, probably 2 years ago.
Yeah, I think so.
But something interesting has happened since. Researchers have stumbled across cockatoos using the water fountains. And
why not? Everyone needs water.
These weren't like motion sensor water fountains. They weren't a simple push. No, it was the spring-loaded twist handle that I remember from like elementary school that has the little knobs on it. You have to turn
Those aren't easy.
No, they are not easy to use. And this particular group of cockatoos are using both feet, as we see in the video that Kiki's playing, to manipulate the twist handle. Then they kind of use their body weight, which isn't a lot because they're a bird. They weigh like a pound, to turn the handle clockwise, keep it from springing back, and then drink water. Around 50% of the time, this is successful. So it's not guaranteed successful, and it's not easy, as Kiki was saying. It's not an easy thing to do.
They have to work for it.
Yes. What's crazy is a lot of them are using it as their primary source of water, and that they will queue up to use it. They will wait in line. This is what we're seeing in this video right now. They're waiting their turn, which is a whole nother thing that they don't even talk about in this article, that like, they're waiting. They're not fighting.
They're not fighting. No. It's like, okay, I'll wait. It's my turn next.
They're waiting in line to use the water fountain, which is more complicated than freestanding water. So like, it's got to be fun. It has to be fun. That is the only thing that I can imagine here, is that it is enjoyable for them to manipulate this water fountain.
I wonder though. I mean, it's enjoyable, but it's also, I don't know. They're social. This is a water source. This is Australia in this area is probably very dry, especially in the summer. I'd love to understand whether the queuing, the lining up is normal in the summertime, or if it's more in the winter when water's more available elsewhere.
So this hasn't been a full research paper or anything. This is very anecdotal, but it looks like they do it regardless of if there's other water available or not. So their other theory is that maybe the water tastes better because it's filtered.
Oh, come on. Yeah, it's filtered. There's no, it's people water. Oh my gosh.
Yeah. So, and parrots do
Not
have a
pond
pretty
water or
good
pool water.
taste. They have a pretty good sense of taste. So I believe that for sure. But I also think the way that parrots are, I think it might be
Smart.
fun. They're smart. It's making them use their brain. It's a puzzle. Parrots like to solve puzzles for fun. So I don't know. I kind of think it's like, oh, I can get my water this way, and I get to like do this fun toy to get my water. Okay.
But at the same time, we see parrots fight over toys and fun.
Also that, yes.
It becomes a wrestling match. It becomes a whole thing. Yeah, so I think the discipline to sit and watch and wait their turn, I think is fascinating. I think it's important. Some of the researchers need to be thinking about, why would birds wait? Why did birds create their own queue? Like,
yeah, truly wild.
I thought that was a people created thing for like elementary schools and the DMV, and I don't know. You want a tiny violin, Blair?
Of course I do.
If you could play the world's tiniest, smallest violin, who would you play it for?
Myself. Obviously. Isn't that the point of the small violin? I'm so upset.
You are so. I play this violin for you. It's tiny. You can't see it. Yes. Anyway, researchers at Loughborough University have just published their work in creating the world's smallest violin with nanotechnology, nanolithography. So technically, they didn't like build a tiny violin, but they did create a lithograph using nanotechnology to create a little tiny violin that is so much smaller than a human hair. 13 microns wide, 35 microns tall. This, what they believe is the world's smallest violin, is small enough to within the width of a human hair.
Joke's on them. I made one that was half the size of a human hair, and I didn't tell anybody.
I just, because I played it and it was gone. It's made of platinum, and a human hair is typically 17 to 180 microns in diameter. Tardigrades are between 50 and 1200 microns. So this is much smaller than a tardigrade. And this was a test project. So what they wanted to do, create this nanolithography system that would allow research projects to develop next generation computing devices and identify new materials and methods that would support that. So the researchers say, though creating the world's smallest violin may seem like fun and games, a lot of what we learned in the process has actually laid the groundwork for the research we're now undertaking. They're doing experiments, probing materials, using light, magnetism, and electricity, and seeing what happens. And so at the nano scale, once they understand how these different materials behave, they can figure out whether or not they can utilize them for computing efficiency, energy storage, harvesting, etc. But anyway, why a violin?
For the title.
Yeah, pretty much. They figured, I mean, if we're gonna do something, we're gonna make it really small, and oh ha ha ha, tiny violin. The entire laboratory, the nano Fraser. It's a nano sculpting machine. It uses thermal scanning probe lithography. And so basically it's like a little teeny tiny, not a laser etcher, but there's like a needle, like that writes the patterns. A little nanoscale needle tip that etches patterns in the nanoscale. Yeah.
So also, to be clear, this is not a tiny violin. It is a tiny image of a violin.
It is. It was a needle etched image of a violin that is smaller than a human hair.
Because I of course I didn't expect it to play, and I didn't expect it to be 3-dimensional. I understand all those things, but I thought it was like cut out in the shape of a violin. It was the shape size of it's etched. So it's basically there's this teeny tiny tape that's like 20 microns wide or something like that. And so there is an image etched on this tiny tape.
Yeah. So, but it's, it's like a computer chip, but there's no chip stuff on it. It's a little teeny tiny violin. So a little layer of platinum got deposited on the chip, and then that etched cavity was filled with the platinum, and lo and behold, you have a platinum tiny violin.
Even better.
Yes, it is. Apparently, it took about 3 hours to create this violin once they figured out how to get it to work. You can't see it without a microscope. And here's a picture, and I know that people in the audio podcast will not be able to hear this, but there is an image of how the tiny violin appears next to a human hair.
That's cool.
Little tiny etching of a violin, tinier than the hair.
Is that blonde hair or brunette hair? Because they're pretty different.
Well, I don't think it's curly.
Fair.
It's pretty straight. It's not.
I just remember in, high school or college or whenever, taking one of my hairs and taking my blonde friend's hair and putting them next to each other under the microscope, and it's pretty, like you were saying, it's a difference of
thickness,
almost 10 times in thickness or something like that. It's crazy.
Yeah.
No, not 10. What did you say?
50 to 180, I think was the
okay. So three in some times.
It depends though. Like some people have thick hair, some people have thin hair.
Yeah.
And curly hair, brunette, there's all sorts of things in there. The science of hair is fascinating. But anyway, this was about a tiny violin that doesn't play for you or anyone. But it's just a picture. Everyone, thank you for joining us for This Week in Science. We have finished our first bout of stories, and I just want to say that I really appreciate you being here. And if you love TWIS, please make sure you get friends to subscribe. Make sure you are subscribed wherever it is that you need to get your podcast or your video streams. We love to have you as a subscriber. If you can head over to TWIS.org, click on our Patreon link, you can become a member of our supporting community. Any amount is great. Honestly, any amount you can give helps us keep going. And $10 or more a month I will say your name, sometimes mispronouncingly or stutteringly, at the end of the show, and I cannot wait to read yours. Please add it to the list. Come on, do it. There's also a Zazzle link at twist.org, and that is where you can buy cool Twist merchandise. So you can have a t-shirt or a hat or a mug or tote bag or a towel or even a bolster pillow. There's some really cool Blair's Animal Corner art projects that are shirts and all sorts of things over there. It's amazing. Blair's done some amazing work over the years. Go check it out. Twist.org. Click on the Zazzle link. Help support Twist, because we really can't do this without you. Thank you for your support. All right. It is time right now to come back to that time in the show. What time is it? Oh yeah. It's time for Blair's Animal Corner.
She loves our creatures, great and small. Biped, millipede, no pet at all. If you want to hear about animals, she's your girl. Except for giant pandas and squirrels, there's Animal Corner.
What you got, Blair?
I have baboons. Did you know that baboons often travel in structured line formations known as progressions as they move through their home range?
I did not. Tell me more.
Yes. They are organized. There seems to be a rhyme and a reason to their order. And there have been lots of different studies trying to explain what the order means and how they come up with the order and why there's an order. And they're all different conflicting explanations. Some propose that the order's random. Some propose that they position themselves with vulnerable individuals in the middle. But there's really not been any conclusive proof for anything. The one with the vulnerable individuals in the middle especially is strange because that's really an herbivore, like a hooved animal thing. Like that's something an elephant might do or an antelope. It's not really something you'd see a primate do very often. So using GPS tracking, researchers from Swansea University re-examined the behavior in a group of wild chacma baboons on South Africa's Cape Peninsula. What they found was that their movement patterns were driven by social bonds rather than survival strategies. And at first mention, you're like, okay, sure, that makes sense. They're primates. But it's not really something that we see in the animal kingdom in animal movement in this particular way, where there appears to be a very clear order to how the baboons go. There's consistency, but that ultimately it's just go find your friend and walk with them. So they tested for 4 different potential organization methods. 1, protecting the vulnerable. So I mentioned that before. 2, competing for resources. That would be the most dominant in the front so they could get to the resources fastest. 3, following the leaders for group decision-making reasons. The dominant is in the front to kind of call the shots for the group. Or 4, patterns emerging from social relationships. And that's the one that turned out to be the correct one. They actually call that social spandrel hypothesis. I'll explain what that means in a second.
I love a good spandrel.
So you know what a spandrel is? I'd never heard this word before. So I learned about it through this study, which is really interesting. So they found that baboons' movement were driven completely by social relationships. The consistent order really was just, where is my friend? And that they all kind of drifted into similar spaces at the same time. The first thing I thought of was the kids who sit at the back of the class to be with their friends.
Exactly. I'll sit back. I'll just drift back here. I don't need to.
Yeah, and then there were people like me who always sits at the front, and I would sit at the front with my friends, and all of us front sitters would sit together. And anyway, I host a science podcast. Is that really surprising to anyone that I sat in the front of the classroom? In the
now a
baboon
new
group,
trivia question.
yeah, exactly. Yeah. That's true today too. When I go into large meetings in the office, I always sit in the front. Anyway, in the baboon group that was studied, the more socially connected, higher ranking individuals walked actually in the middle of the group, while lower ranking baboons were in the front or at the rear. And that is because they knew where they were going. They didn't need a leader. But they also weren't worried about vulnerable individuals in the middle. Like, they were just kind of going. Everybody knew where they were going. They just sat where they wanted to sit, sort of thing. And so this introduces this concept of a social spandrel. So, Kiki, what's a spandrel?
A spandrel is an evolutionarily very interesting and confusing kind of thing.
Yes. So you're correct. This is what I learned, is the word is actually an architectural term.
Yes. It's like a flare. It's like, where's your flare?
Yes. Yeah. It is the triangular space that emerges as a byproduct of arches placed side by side. So it's the flare in between two arches that doesn't have any function.
But it does, because if that weren't there, then the arches, or if it weren't the flare that it was, the arches would fall down.
Right, but it's there because the arch is there. And so the arch has been designed and erected in a particular way. And as a result, there is a negative space that looks a particular way. And so in biology, a spandrel is a trait that arises because it is a side effect of something else that was selected for. And so in this case, travel patterns among baboons emerge naturally from social affiliations and are not a strategy.
So baboons terrify me. I think they're beautiful.
Yes, as they should. Yes.
They're terrifying. And so to find out about this, they line up like the cockatoos for the water fountain when they go for a walk. It was a little bit like spiders, cooperating. The kind of, are they like, is this a strategy? What are they doing? How are they why are they doing it like this? I was very concerned for a minute, so I'm glad it's a spandrel.
Yeah. They're just hanging out with their buddies, which of course there is an evolutionary impact to that. There is improved reproductive success and survival rates amongst baboons with strong social bonds. So reinforcing those social bonds is an evolutionary advantage. So just hanging out with your friends on the long walk is an advantage, but the order is a complete byproduct. So there you go.
Cool. So it could be anybody in a certain order, depending on where your friends, where you all kind of end up in that walk.
So spandrel, social spandrel. Anyway,
I think a lot of people here just learned about social spandrels. That's the word of the day, everyone. Social spandrels.
Yes. And moving from social spandrels, to climate change, I have a study about birds and climate change. So we talk a lot about how plants and animals cannot escape climate change. But usually the one animal that could is birds, because they can fly. Their entire thing is flying south for the winter, escaping the temperatures they don't like. Right. And so there's a narrative that it's like trees can't migrate, turtles can't migrate, slugs can't migrate, birds can migrate. A new Yale study actually looked at the escape abilities of birds from a warming world. And unfortunately, the news is not good. Research tells us they cannot move fast enough or far enough to keep up with climate change. That's essentially the whole story, but I'll give you a little bit of data. Yeah.
But like trees, we've been talking for a long time about climate change and how it's moving faster than species are able to. And this has been a big question about birds, the plant species, the insect species that they feed on, that they nest in, that is it the ecosystems that are impacting the speed of the birds? Or is it
good question,
the birds limiting themselves?
Right. So we don't know. Also, Doesn't matter because
I mean, in the end, it doesn't, but it's the kind of thing
Yes.
where if we could influence it in some way,
right.
like
No,
plant-
and for that reason it does.
can we move
but
an
yes,
ecosystem? Like
the
you know,
question
the
is,
amphibians.
can the birds fly farther than they're flying? But they're not because their niche is not following them. So if they flew to a place they've never been before, there's no familiar food, there's no familiar habitat, there's there's unfamiliar predators. You're out of luck anyway, right? So that's a really important point, is even if birds could escape climate change, would they ultimately perish anyway because they're being thrust into a completely unfamiliar space, right? So really it's how fast can the trees move, and that can often be a limiting factor. So the data is supporting that either they're choosing not to go as far or they physically cannot move as far. They studied the movements of 406 species of North American birds. This data was collected by citizen observers over 2 decades and corresponding local temperature changes. During the summer, bird species on average relocated between 40 and 50 miles north during the period covering the data. And sometimes they relocated to higher elevations as well, which, as I used to teach in my Zoomobile program about climate change, when you use elevation as an escape from climate change, there's an inherent problem in that you have less space to move than if you're moving latitudinally.
Yes.
So you can go north and north and north until you hit the North Pole. But if you're on a mountain, you can only go so far up. And you're stuck.
For many reasons. Yeah.
Yeah. So, anywho, a lot of these bird species relocated. On average, the northbound movement helped birds avoid a temperature increase of about 1.28 degrees Celsius.
That's a lot.
But that was only about half of the temperature increase they would have experienced if they stayed. So that means on average, they still experienced a 1.35 degrees Celsius increase in temperature during the summer months compared with the temperatures in their original home range. So they still had a 1.35 degree increase even with their movement. So they didn't outpace climate change. Climate change still caught up with them, even though they were adjusting their territory. During the winter months, birds had only minimal success in limiting exposure to warming. They experienced only an 11% less warming than had they not moved at all. So winter, by and large, was unsuccessful for them to relocate. In the winter, birds experienced on average 3.7 degrees Celsius increase in temperatures over the 20 years. That's huge. So overall, more than 75% of birds managed to reach slightly cooler climates, but some species like the cactus wren, which is from deserts and arid systems in North America, didn't move at all. So they were susceptible to climate-triggered changes in the environmental niches. So like a desert bird didn't move. That's
because
bad.
it's hot. The desert is already very extreme. And
right,
so
but if you see like the videos
that
coming
bird
out of
gonna
Death
go?
Valley in the summer, still bad though. Like even if you're used to the desert, it's bad now.
poor little birds. Cactus wren are so wonderful, and they have no, there's canyon wren. Cactus wren are different, but the canyon wren. But
Well, all wrens are
All of
actually
them.
really cute, I think.
I love wrens. They're just the cutest little. They're like the mice of the bird world.
Yes. Oh man, that's really good. Yeah, so by and large, the bird species capable of flying long distances were the best at limiting their exposure to warm climates. So that follows the hypothesis that capability, like physical capability of movement, does factor in, because
Yeah.
the ones that naturally fly farthest were the best at relocating. Okay.
Makes sense.
But species that were less mobile they didn't go as far. And so, you know, they really had to, they had similar movements to reptiles and mammals in the area. Actually, they weren't that different, even though they had wings.
So there's a lot of research in the bird, like nomadism, like migration, or the birds that just stay home. And what they have found is that any bird can just stay in one place, right? You can be that. But the birds that are more likely to migrate are actually more usually generalists, and they have larger habits, and they have those larger flight abilities. They have figured out how to adapt to changing food availability, and they, instead of staying in the same place and like a mountain chickadee maybe, or black-capped chickadee storing nuts for the winter, they're like, I got to get out of here, and they fly away. And so the specialists are the ones that are in trouble, because they're the ones that kind of stay in one place because they're specialized, and that is like, they don't fly as far. They're usually in that one little place, and that's like, even though they have wings, they are so specialized to their niche. They're
creatures of habit. Yes.
They're not going to go anywhere. And they're the ones, like people who are like, you'll have to pry me out of my home, the
ones that ignore the flood warning and then end up on the roof. Yeah.
Yep.
Yes. So the researchers draw kind of a larger conclusion that this is very concerning, because even these animals that have the physical capability to move out of these climate impacted areas are not able to move as much as we would expect. And so, of course, that means that less mobile species are obviously at a large impact as well. And so,
because how are they going to, they can fly a little bit, but when are they going to be like, oh, I got to get my energy and then fly a little bit further and get my energy and fly a little bit further? They still have to do that adaptation. They have to do that population level change, which it's not going to happen fast enough.
And if you're a lizard or a mouse or a tree, you're really in trouble. Yeah. So it does change the picture of climate change and the movement of species, that birds are not this kind of like get out of tree free is what I was going to say. That's incorrect. They don't have a free ticket to get out of the climate change impact zone, basically. I wish more complicated.
Some species did. Some of them do, but definitely not all of them. It's going to impact diversity.
But, might as well cut all climate change research funding. Who cares?
Yeah, right now. It's time. Let's do it. Yeah, sure. Anyway. Oh, my God.
Kiki, you had wanted to talk about chimps last week. Do you want to introduce this last story?
No, it's your animal corner.
Oh, okay. All right, I'll do it.
It's fine. Yes, it's yours this week. I was like, I hope Blair brings it next week. Oh, my gosh. Yeah.
Perfect. Yeah. Hey, chimps play the drums on trees. We knew that already.
News at 11.
Yeah, no, we talked about, again, I don't remember how long ago this was, but we did talk on the show about how chimps kind of drum with their hands on tree bases, and the research concluded that it was actually a long distance communication method, which is insane in and of itself.
It just boggles my mind. Yeah.
But this new study, this is a 5-year study. They looked at video footage at 5 distinct locations where there were chimp communities, and they found that they were striking trees with rocks. It was adult male chimpanzees. They were repeatedly hitting the tree trunks with rocks, and there would end up being these piles of stones at the base of trees. They're like throwing these rocks at the bases of the trees. And they called this stone assisted drumming. Now, what's interesting is the previous behavior that they documented, where they were hitting it with their hands, they were very quiet before and after. But for this one, they got loud and like raucous, and they like hyped themselves up before they threw the stones. And so their theory is that this is meant to carry further. So this is their way of kind of making a long distance phone call. I don't know. Maybe they're just psyching themselves up before they throw the rock so they can throw it really hard. Kind of like,
yeah, I mean, I'm going to do it. I'm going to do it. I'm going to do it.
Yes. Yeah.
Yes. And then you go for it. But they didn't, they found that there were like certain patterns they were doing and things that it's not just throwing rocks at trees, that this is a form
of communication. Yeah.
Yeah. And that to me is the wildest part of this, that these chimpanzees not just are, maybe close up, drumming a little beat, but using these trees. Yep, there was that.
Terrifying.
Throw a rock, kick the tree, but you can hear that bass in the trunk of these massive trees. Dong, 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 dong. And these root systems penetrate the earth, and there's probably some aspect to this that other chimps are very interested in.
I think it's interesting that these trees can take this beating also. There are some trees that I've seen, particularly in North America, that could not handle this.
But I'm sure that this is not all trees, right? This is trees that can take it, and the tree in the video that goes along with this story has definite wound.
Yes,
it has been
big old wound. Yeah,
but it's a massive tree. So, that too it could be like a marker, a territorial display. Like there's so much that could go into this, but yeah. The sound.
Yes, and
if a chimpanzee is attacking a tree, you don't want to be anywhere near it.
No. You don't want to be near chimps in general.
Anyway.
But they're calling this a cultural transmission because young chimps adopt the behavior from older group members, and particularly when there is something deemed as communication that transmits from generation to generation, that is considered social learning, that is considered culture. When you're talking about animal behavior. And so this is a pretty complicated and a pretty specific thing that they are communicating down through generations of how to communicate with each other long distance. So it's chimp culture via rocks at trees.
The learning.
Bonk. What could you possibly be trying to say?
I don't like the ketchup here. But
when are you coming home? Later.
The distance where you have, exactly. I'll come home later. Hey, I saw another chimpanzee over here. Oh, there's a squirrel. Or like, what? This is the thing. There are so many bits of information being shared that we do not understand yet. And I think it's fantastic.
Yeah, that's what I want to know. What are they communicating via these roots?
I mean, if it's important enough for the young chimpanzees to pick it up and transmit it themselves from generation to generation, then this is something that is important socially. Might be a spandrel, but it is. It's socially important. So that's. I think that's it. That's fascinating. And what I'd like to know is how does it differ between different little tribes of chimpanzees? Do different tribes talk to each other this way, or is it just within group? Like, what's going on there? I think there are some interesting questions to be raised. I don't know. Chimp rock. It is. It could be that would be that ad from like the 90s, 80s. Hey, what was that ad? It was like, whatever, what is that? It's whatever rock. Whatever. It was like you'd order all the cassettes in the mail.
Oh, sure. You're talking about
somebody in the chat room knows what I'm talking about.
Oh my goodness. Yes, it's the you pay a penny and you get like 100 CDs. Yes, I know what you're talking about.
Yeah, it's Planet Rock. I don't remember what that was, but yeah. Nope, no.
Columbia House.
No, but there was an advertisement that was specifically like the advertisement was like this whole it's blah blah rock. Like it was a very I don't know. Someone will tell me later. Podcast people watching later. Please help me with this old social reference. It was something rock. It was I don't know. No, it's not the label, everybody. It was like this. It was in an advertisement, and it was the line in the advertisement that was said by somebody who sounded like Cheech Marin or something like that. Anyway, whatever. Help me out, everybody. This is going to be. I'll figure it out. I'm going to move into my section of stories with an animal story. Birds, no more chimpanzees. And Blair, honestly, this is my favorite animal story from the last week. Researchers just published in Bio1, which is an open journal online, the first GPS observation of a western gull, Larus occidentalis, riding in a long-haul garbage transfer truck. So, okay, long-haul garbage transfer truck, what's the big deal? They had a tracking device on this western gull in San Francisco. They had a bunch of them on these gulls, western gulls in San Francisco. And so these GPS tracked gulls have been tracked during the breeding season at Southeast Farallon Islands off the U.S. West Coast. Lots of great white sharks out there from 2013 to present. And they do foraging trips from the Farallons to land, and they tend to be longer than at-sea trips. And so where do the gulls go? Very often they'll go get human food. They'll get food from people. And so this one instance, they tracked a little lady, western gull, who got in the back or whatever, rode a long haul garbage transfer truck, an 18-wheeler garbage truck from a waste transfer station in San Francisco, California, which is probably just that garbage, like right near Candlestick Park in South San Francisco. This gull rode this garbage truck to the Central Valley, California, to an outdoor compost facility. And then she spent a day there and flew back to the Farallon Islands, went back to the garbage truck, rode it again. So May 21st and May 23rd, this female western gull got on an 18-wheeler garbage truck to a Central Valley compost station to go eat. So she flew from the Farallon Islands to the garbage, got on a truck. The truck drove her to a massive food source. She did what she needed to do there, flew back to her babies at the breeding grounds in the Farallons, and then went back again. She's like, oh yeah, that was great. I didn't have to fly. That was awesome. And so these foraging trips were longer than average. So by about 14 to 18 hours longer. And she went 95 to 130 kilometers farther from the colony than most birds do. And this is further than this individual's other trips during the week-long tracking period. And they haven't really noticed this in a lot of gulls, but they say it's not been widely observed, which leads me to believe this might be something that other birds are picking up. And so I hope to hear about it. And so they don't understand the bird's intentions. And honestly, I think energy is a big deal. Like, oh, how did she find out that if she rode the truck, she'd end up in a compost facility and then she would be able to fly home and she would have half the journey, more of the food. I mean, this is efficiency optimization at its finest. This is amazing. So the next time you see a Western gull in a human garbage dumpster or on a beach or wherever, just give them a nod and say, yeah, I know, you're smart. This bird figured something out. It's really amazing. Yeah. So the question is, will others learn it? And we don't actually know that. So I find this one of the most interesting stories of the week because it's a bird, and everybody thinks that gulls are just kind of like the pigeons of California, but gulls are amazing species of birds. They are diverse and widespread, and very often not necessarily coastal. And so gulls are generalists in terms of species that are able to adapt and find new food sources and create new behaviors like this one. Yeah, they have long developmental periods. So a lot of gull species will not actually be mature until their fourth or fifth year. And so they'll be with their parents for several years before they're actually off on their own as breeding adults. So gulls are actually very interesting. And so when you say seagull, it's not just a seagull, it's a gull, right? Okay, so I'm giving you birder geek advice. So we're going to move on from the first time anyone has seen a gull riding a truck to a compost station to a question that I find actually very interesting, which is what is it in the brain, in the nervous system, that creates what is called the myelin sheath and what impairs it from being repaired in situations like multiple sclerosis. And so research out this last week published in Neurology, Neuroimmunology, and Neuroinflammation. It's a study that's called Pro-Inflammatory Molecules Implicated in Multiple Sclerosis Divert the Development of Human Oligodendrocyte Lineage Cells. So the interesting thing about this study is we know that in multiple sclerosis, the myelin sheath, which is around the axon or the transmission part of neurons within the nervous system, that becomes damaged by the immune system. The autoimmune system gets dysregulated and something happens. What's going on? Oligodendrocytes, they are a type of glial cell, which is not a it's not a neuron, but they're one of the support cells. Oligodendrocytes are one type, and there are astrocytes, oligodendrocytes, there are a number of different glial cells, and they have different functions. This particular one, it wraps the axon, becomes myelin, which is supposedly important for conductance and all this stuff. Anyway, it gets damaged, and then there's scar tissue, and they don't get fixed. But why not? What could fix it? And this study is fascinating because they were able to determine that the inflammation that occurs, this dysregulation to the autoimmune system, leads to an environment in which if you put new oligodendrocytes, little baby oligodendrocytes that could repair the myelin, become new myelin cells, they're supposed to grow into oligodendrocytes. Those glial cells in the inflammatory environment that is multiple sclerosis, they turn into astrocytes instead. So there are signals involved that actually stop the glial cells that would become myelin from doing that. And they go off and they become these other kinds of supportive cells. And so what this study suggests is that if we could actually figure out what that signature is, what the cell signaling molecules are, that we might be able to downregulate whatever it is, control it, and stop this differentiation into astrocytes, and maybe turn the cells into myelin, actually, which would be really amazing. So there's a basis, a potential for being able to repair myelin that is natural and that it's part of what the body's doing, but it has to do with reducing the impact of inflammation on these particular cells. The other aspect of the study that I think is really interesting is just the pathways that get shifted. So you pull a lever, you go down a different track, right? You get switched from one track to another. And that this is the kind of thing that inflammation can impact and control, so that if you have a stressful environment, if you have autoimmune disorder, whatever it is, where an inflammatory environment is the norm, then different cellular decisions are being made than ones that would be more supportive for long-term survival. So I think this is a really interesting study. I think it has implications for a lot of things beyond just oligodendrocytes, astrocytes, and myelin. But for sure, they specifically may be able to go down this path to support therapy for multiple sclerosis, which I think is absolutely fascinating. Blair has run off somewhere for a moment, and I have one last story so that we can finish off the show. And I was hoping that she would come back so that I could tell her not to tape her mouth at night. What? What am I talking about? Apparently, there is a huge trend on TikTok where people in videos are taping their mouths shut at night to keep from getting wrinkles, or to help themselves with jawline definition, energy levels, sleep apnea, oral hygiene. Anyway, it is a viral phenomenon where people are taping their mouths at night because influencers on little video platforms are saying it's good for you, with absolutely no support. So you know what? People have studied it. Apparently there's a few studies, right? And so yay, this week, scientists from London Health Scientists Center and the University of Saskatchewan College of Medicine just published a study where they reviewed a bunch of those studies. And oh, guess what? They're not very good studies. Great. The data, they looked at 10 of the most relevant research papers, 213 patients in total, to even look at what was going on with this hyped, I don't know, self-improvement therapy. Anyway, the studies were looking at how the mouth taping might influence things that people wanted to be treating: sleep apnea, nasal obstruction, mouth breathing, jaw definition, etc. And in them, of course, these people were monitored in controlled laboratory environments. In 2 papers, they did actually see a mild improvement in patients with mild sleep apnea. And so this was basically how many times did they kind of stop breathing during sleep, when was blood oxygen saturation dropping during sleep, but it was not really significant. So there was a little bit there. And really, the researchers write in their paper, which is the thing that I think is the most important. Studies showed that mouth taping offered no differences and even discussed potential risks, including asphyxiation in the presence of nasal obstruction. And again, I quote There are potential serious detrimental health outcomes to those with nasal obstruction who seek oral taping as a means to ameliorate their mouth breathing, obstructive sleep apnea, or sleep disordered breathing during sleep. And really, you're sleeping, everybody. Come on. You're sleeping. And here you are. Here you are. Sleeping. And then you tape your mouth, and the only thing you can breathe with is your nose. And if you have allergies, if you have any, please don't do this. Okay? Don't do it. This is not the way to do it. All right? I'm just saying it right now. The conclusion from the abstract of this paper, the social media trend of mouth taping for individuals with mouth breathing, sleep disordered breathing, or sleep apnea has been reviewed based on the data presented by these 10 different studies. It seems that there's a potentially serious risk of harm for individuals indiscriminately practicing this trend. Further studies are required to elucidate any clinical benefit this practice may have. So, everyone, I'm just gonna ask you to not just. And these aren't like cinnamon challenges or whatever. It's like, oh, you're gonna help. This is gonna help you. TikTok is not a place to do research. Okay, like. I'm really, really concerned for people in the world today. I really am. Oh, influencers, thank you very, very much for creating a need for researchers to actually discover whether or not taping your mouth shut while you sleep is advisable. Blair, are you going to come back for a second for the end of the show? Are you done? Oh, it's the end of the show. I think I've done it. I think I have done all the stories. And Blair doesn't have any more stories. So I don't know. I'm guessing there's a child. Oh, Blair cannot. I didn't know there was a heart out. I'm guessing it was a child. So on that note, thank you for answering the question, Blair. Thank you for being here tonight. And everyone, thank you for joining us for another wonderful episode of This Week in Science, talking about all the science that we declared fit for discussion for this week. Really appreciate you being here. I've got to give some shout outs to everybody tonight. Shout outs to everyone in the chat room right now, wherever you are. Thank you for being here, for chatting and being a part of this conversation. It's always so fun to be able to see your comments and your questions and be able to incorporate them, because this is a whole conversation. We are a community. Fada, thank you for your help with social media and show notes, especially last week at the last minute cancellation. Gord, Arnlore, thank you for manning these chat rooms, helping to keep them great places for everyone to hang out. Identity 4, thank you for recording the show. Rachel, thank you for editing the show. And I would like to thank our amazing Patreon sponsors for all of their help supporting us on Patreon. Thank you to Robert Norlin, Robert W. Farley, Lauren Gifford, Dana Lewis, Eden Mundell, Ali Viola, Aaron Anathema, Arthur Kepler, Craig Potts, Mary Gertz, Teresa Smith, Richard Badge, Bob Coles, Ken Northcote, George Corris, Pierre Belazar, John Ratnaswamy, Chris Wozniak, Vigard, Chefstad, Donna Styles, a.k.a. Dunstylo, Ali Coffin, Schubert, Don Menace, PIG, Stephen Alberon, Daryl Myshak, Andrew Swanson, Fredis 104, Sky, Luke, Paul Brunovis, Kevin Reardon, Noodles, Jack, Brian Carrington, David E. Youngblood, Sean Clarence Lamb, John McHugh, Greg Riley, Mark Hessenflow, Steve Leisman, Ike Azima, Ken Hayes, Howard Tan, Christopher Rappin, Richard Brendan Minish, Johnny Gridley, Remy Day, G. Burton Lattimore, Flying Out, Christopher Dreyer, Artyom, Greg Briggs, John Atwood, Rudy Garcia, Rodney Lewis, Paul Flip, Shane, Larson, Craig Lander, Sue Doster, Jason Olds, Dave Neighbor, Eric Naplon, Makes, E.O., Adam Mishkan, Kevin Parajan, Bob Calder, Marjorie, Paul D. Disney, Pecoraro, Tony Steele. Thank you for all your support on Patreon. Can't do it without you. And to everyone out there, if you would like to support the show, please head over to twis.org, click on that Patreon link and become a supporter on an ongoing basis. Help keep curiosity and science within our minds. Help us do this. Also tell a friend, right, about TWIS. Anyway, we will be back next Wednesday, 8 p.m. Pacific time, broadcasting live from Twitch, YouTube, and Facebook. If you want to listen to us as a podcast, search for This Week in Science wherever you find your podcasts. And of course, like I said, get your friends to subscribe too. It's more fun when there are more people. For more information on anything you've heard today, show notes, links to stories, go to twist.org. You can also sign up for our newsletter there. I keep thinking about it. There might be one. I just sign up. It'll be great. We don't do anything. This is a safe email list. I'm not sharing it with anybody. We love your feedback. If there is a topic that you want us to cover or address, let us know. If there's an interview, whatever, let us know. Send us an email. Talk to us on our social media accounts. Put TWIS in the subject line if you're going to be emailing us so that your email doesn't get spam filtered into, oh goodness, the gullet of a gull who's on a garbage truck heading out to that Central California compost facility. Good grief. Never find it. We look forward to discussing science with you again next week. And if you have learned anything on the show tonight, remember, it's all in your head.
This Week in Science. This Week in Science. This Week in Science. This Week in Science, it's the end of the world. So I'm setting up shop, got my banner unfurled. It says the scientist is in, I'm gonna sell my advice. Show them how to stop the robots with a simple device. I'll reverse global warming with a wave of my hand. And all it'll cost you is a couple of grand. 'Cause this week's science is coming your way. So everybody listen to what I say. I use the scientific method for all that it's worth. And I'll broadcast my opinion all over the earth. 'Cause it's This Week in Science. This Week in Science. This Week in Science. 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 This Week in 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 Science.